Good day, everyone. Today, I'm delighted to present my research results in this prestigious conference on the exciting topic of adding colchicine to doxorubicin for better anti-tumor efficacy and thus associated toxicity in an in vivo cancer mouse model, a step toward their combined use clinically. Our agenda consists of a brief introduction, followed by our hypothesis, then aims, then the methodology, then we'll discuss the results, and finally we'll state our recommendations. Doxorubicin was originally isolated from Streptomyces pyocetus. It's a potent anthracycline chemotherapeutic antibiotic drug effective in the treatment of several cancers. To make it crystal clear, our problem is that although doxorubicin is a highly potent chemotherapeutic drug, it's highly toxic that patients that are cured of cancer are prone to death from other problems. In addition, cancer cells start to develop resistance. So how to address this problem? We try to give a hand by adding colchicine. So why colchicine? Colchicine is a widely available, cost-effective, and already FDA-approved drug of a natural origin. It was originally isolated from Colchicum autumnale. It's a tricyclic alkaloid drug. It's clinically used in inflammatory diseases as gouty arthritis and familiar Mediterranean fever. This study aimed to investigate whether or not colchicine can enhance the anti-tumor activity of doxorubicin, determine the effect of colchicine alone or in combination with doxorubicin on cell apoptosis, cell cycle, and cell proliferation, investigate whether or not colchicine can ameliorate the side effects of doxorubicin while using lower doses. Now let's delve into the methodology where we will talk about the drugs and chemicals, followed by the experimental design, then the analysis done where all statistical analyses were performed using GraphPad version 8. As for the drugs, we use doxorubicin as androgen, we used colchicine from Nostro Company, and all chemicals were of high purity grade. As for the experimental design, the Research Ethics Committee at Faculty of Pharmacy and Chams University approved our protocol, and all animals were treated according to the ARRIVE guidelines. First, we obtained stock mice bearing early cells intraperitoneally from the National Cancer Institute, and we extracted the acidic fluid, diluted it, and adjusted the count of the cells to 250,000 cells, inoculated this count in 6 groups from group 2 to group 7. Group 1 served as a negative control group. Group 2 served as a mock treated group. Group 3 was given doxorubicin in its high dose 2 milligram per kilogram intraperitoneally on days 3, 5, and 7 of tumor inoculation. Group 4 was given colchicine orally on days 3 and 5 and 7. Group 5 was given both dox high dose and colchicine. Group 6 was given doxorubicin in slow dose 1 milligram per kilogram, and Group 7 was given both doxorubicin low dose and colchicine on the same treatment days. On day 8, half of the animals in each group were sacrificed to obtain the ascites. Blood samples also in the liver was extracted for the different analyses. Half of the animals were left alive to determine the survival along 24 days of tumor inoculation. As for the analysis done, to assess the anti-tumor effect, we calculated the percentage of body weight change. As for the extracted acidic fluid, we measured its volume, determined total tumor cell count, live and dead cell count. The survival was monitored along 24 days. Also, apoptotic analysis of early cells was performed using an XMV and propodium iodide stains. As for the hematological analysis, we assayed the CBC of the mice. Moreover, cell cycle analysis was performed using cycle test plus DNA kit using flow cytometry. We also assessed the inflammatory and oxidative stress markers as interleukin-1 beta, CRP, superoxide dismutase catalase, and MDA. We also assessed the liver and kidney function test. Moreover, we assessed the lipid profile. As the effect of the treatments on the body weight change, inoculation of early tumor significantly increased the final body weight when compared to the control mice. All the different treatment decreased the body weight significantly when compared to the control group, where dox high showed the highest reduction effect on the final body weight. Dox high dose even led to a lower final body weight than the control mice, which could be due to the effect of doxorubicin on the tumor cells as an anti-cancer and side effect as of decreasing the body weight as previously reported. As for the effect of the drugs on the different tumor parameters. All the different treatment groups decreased the tumor parameters as shown the tumor volume, tumor total cell count, living dead cell count, where doxy and colchicine showed the highest effective reduction concerning the tumor parameters 18.3-fold and 37-fold. We can also conclude that colchicine increases the doxorubicin-induced anti-tumor effects when comparing doxy-colchicine to doxy-solid treated group, solid treated group, or doxlo-colchicine versus doxlo-solid treated group. The survival of the mice was monitored along 24 days of tumor inoculation. On day 12 of tumor inoculation, 100% of the untreated mice were dead. Colchicine slightly extended the survival until day 17. Dox-high treatment extended the survival till day 24, where 100% were alive. Dox-low extended the survival till day 24, where 57% were alive. So colchicine slightly enhanced the survival, but only slightly when compared to the treatment with dox high or low. Combination therapy extended the survival till day 24, where only 85% were alive. We can conclude that doxorubicin and colchicine extend the survival of early cells. Representative flow cytometry charts of annexin V promodium iodide stained cells are shown here, where the percentage of early and late apoptotic cells in dox-high, colchicine, or dox-low, or their combinations, was significantly increased compared to the earliest control group, contrary to our expectations. When comparing dox-high colchicine treatment group versus dox-high solely treated group, colchicine addition did not affect the percentage of early and late apoptotic cells significantly. This could be due to the complete curing effect of the high dose. However, concerning the low dose, when we combined it with colchicine, there was significant increase. Addition, impact of the co-treatment on the cell cycle analysis, doxorubicin arrest was found to arrest the cell cycle and the S phase, which is in line with its mechanism of action as it binds to topoisomerase 2 and inhibits DNA synthesis, whereas colchicine arrests the cell cycle and the G2M phase in line with its mechanism of action as binds to tubulin and inhibits mitosis. As for the effect of the drugs on the hematological parameters, inoculation of early tumor-induced hematological toxicities as shown by decreased red blood cell count, hemoglobin content, and hematocrit value, all treatment enhanced these blood indices. Concerning the white blood cell count, it was significantly increased in rich-bearing group due to the inflammation induced by the tumor, whereas it was significantly declined in all other treatment groups. As for the effect of the treatments on the ELTST urea and creatinine levels, tumor inoculation increased the ELTST urea and creatinine levels, indicating hepatic and renal toxicities, whereas all treatment groups decreased these levels significantly. As the effect of the drugs on the lipid profile, tumor inoculation increased the total cholesterol, triglycerides, LDL cholesterol, and decreased the HDL cholesterol. All treatment enhanced the lipid profile to some extent. Treatment of early-bearing mice with doxy led to a decrease in the serum total cholesterol and triglycerides when compared to early control group. However, treatment with colchicine or doxlo decreased the total cholesterol but not the triglyceride levels. This shows that colchicine or doxlo has potential anti-tumor effect but not as doxy. Treatment with either combination of doxorubicin and colchicine significantly decreased the total cholesterol and triglyceride levels. As for the effect of the treatments on the oxidative stress, tumor inoculation caused oxidative stress as shown by decreased superoxide dismutase and catalase activities and increased MDE levels. Treatment of early-bearing mice significantly increased the sodium catalase activities and decreased the MDE levels in all treatment groups when compared to early-bearing mice group, but not as the negative control group. Interestingly, when comparing the dox-high colchicine treatment group to dox-high solely treated group, the oxidative stress was decreased. As for the effect on the different treatments on the inflammatory markers, tumor inoculation raised the CRP and interleukin-1 beta levels significantly. Treatment of early-bearing mice with doxorubicin high dose further raised the CRP and interleukin-1 beta levels. This could be due to the effect of doxorubicin on the NL or P3 inflammasome pathway. Doxorubicin low dose only slightly increased the CRP interleukin 1 beta levels. In contrast, colchicine decreased these inflammatory markers. Maybe due to the effect of colchicine, inhibitory effect of colchicine on the NLRP3 inflammasome pathway, co-treatment of colchicine and doxorubicin decreased the inflammation when compared to early mice that received doxorubicin only. Either dox high or dox low co-treatment with colchicine enhanced the doxorubicin anti-tumor effect as shown by decreasing the tumor parameters, the tumor volume, tumor cell count, increased apoptosis, increased cell cycle arrest, enhanced survival, and also co-treatment ameliorated the dox-associated side effects by enhancing the kidney and liver function test, enhancing the blood indices and lipid profile, decreasing the inflammatory markers and the oxidative stress. As for our recommendations, the study paves the way for a patent registration by the SCDF project. The study opens a new avenue to colchicine use with doxorubicin in clinical settings after completing other preclinical experiments. This study shed a light on the benefits of drug repurposing instead of synthesizing new molecules. Finally, the study implicated that to overcome multi-drug resistance, we can use combination therapy. This study was published in the Journal of Archives of Pharmaceutical Sciences at Ain Shams University under the honorable supervision of Dr. Muhammad Labib Salem, Dr. Nadia Hamdi, and Dr. Imam Farag in collaboration with Dr. Sophie Van Lent-Out, Professor of Immunocardiology, Berlin Institute of Health. This study was part of a project funded by the STDF. Thank you so much.
Thank you so much, Dr. Mamdouh, for your wonderful talk. We have actually quite a few number of questions that are already popping in. And I really like this theme of repurposing drugs that have already been synthesized and well studied. And actually, a lot of our questions are about the fact that colchicine has been well studied. And so knowing, some of the other effects that we know it has on the body. And so, first things first, I guess, can you speak to some of the side effects of colchicine treatments that we are aware of at this point, in cancer and also in other indications? And do you think there would be any risk of using colchicine for its immunosuppressive qualities?
Okay, colchicine is known to treat familial Mediterranean fever, so it's FDA approved. However, it has some side effects that are tolerable, such as diarrhea and also, there was a case report, actually, I read that a patient with lymphoma who was on a chemotherapy regimen that includes doxorubicin and took colchicine, and he had severe side effects and had to stop the colchicine. So I read a previous case report, so it's a case report, it's not the usual, so maybe we need further studies.
Right, yeah. I think it's especially interesting that this is in a case of lymphoma that you saw that case, given what we know that colchicine can have this effect of reducing the white blood count in patients. So potentially there's some aspect of particular cancers that might be better than others in which to use colchicine. Do you have any comment there?
Excuse me, can you repeat the question?
Yeah, do you have any comment on, if there are particular cancers that might be more amenable to colchicine treatment or multiple drug resistance therapies than others?
Actually, I tested this in mice using early site carcinoma cancer. It's undifferentiated cancer. So, its origin is in breast cancer, but it's undifferentiated, so I cannot tell if it's it has some preferential efficacy on some cancers or not.
Sure. And so one of the other questions that we had is how do you think colchicine will be impacting kidney injury that's mediated by doxycycline?
Oh, I tested the urea and creatine functions, and colchicine had beneficial effects and protective effects on the kidney.
Do you know what the mechanism might be in this case?
Colchicine is an anti-inflammatory drug, Also, it inhibits the NLRP3 inflammasome pathway and decreases the subsequent interleukin-1 beta, interleukin-18, and the subsequent release of CRP, and the subsequent release of interleukin-6. And also, it's antioxidant. It was proved to be an antioxidant drug. So, all these effects decreases the oxidative stress caused by doxorubicin, as the doxorubicin is oxidized inside the body.
I see. And I guess is there any idea that you might have in this case of, combining with other drug combination systems? So for example, doxorubicin with rapamycin instead of colchicine.
Colchicine is already a natural is of a natural origin, so we can think and analyze this. So this is really inspiring. We can
I
test further combinations. Yeah.
Yeah, absolutely. I guess one final question from my side would be when you think about all of these, the side effects that are manageable and then potentially the side effects that are potentially a little more harmful, are there any ways that you think route of administration in humans specifically, do you think there's any way that we can control these off-target effects of colchicine?
Um, oh, actually, colchicine is taken orally in family Mediterranean fever. So it doesn't have much off-target effects. It's the contrary. Doxorubicin has off-target effects, but colchicine is not doesn't have much off-target effects.
All right. Well, yeah, that's great. I think if there are other questions, oh, I think we actually just had another question coming up. So one more question we're wondering about is, do you think there are any considerations out there in the field, in your group or outside in generally the field, of modifying the colchicine molecule further?
Yeah, there are a lot of studies and a lot of papers published in modifying groups and adding some groups in the colchicine so it can fit better in the colchicine binding site and enhance its efficacy. And therefore, we can use lower dose when the efficacy is enhanced.
Absolutely, and then cut out some of those side effects that you were mentioning.
Yeah,
Wonderful. Well, thank you so much for your talk and these very engaging questions.
thank you so
Rana,
much. Thank you so much. I like this conference so much. Yeah, there's researchers from all over the world.